ఇదిగోండి సార్, ఇదిగోండి ఇక్కడ మీరు జరుపుకుంటూ పోండి. ఇక పక్కన స్లైడ్స్ ఉన్నాయి కదా, మీకు దాని నుంచి వేసుకుంటాం. ఇక మీకు అది వస్తుంది. ఇక 1 2 3 ప్లస్ చేసుకుంటా పోండి. ఫుల్ స్క్రీన్ పెట్టుకుంటే పెట్టుకోండి.
సెకండ్ వస్తుంది. ఎక్కడది ఈ సైడ్ ఈ థర్డ్ మీద చేస్తే థర్డ్
ముందు పెట్టుకోండి.
స్లైడ్స్ లో పెట్టేసుకో. స్లైడ్స్ లో పెట్టు. స్లైడ్స్ లో పెట్టు పెద్దది. ఇది దిస్ వన్ అయ్యా.
ఇప్పుడు మనకి ఇది అవ్వకూడదు, నెక్స్ట్ ఇది అవ్వకూడదు
కాబట్టి అదే పెడదాం.
మీకు ఏం అవ్వకూడదు, మరి నెక్స్ట్ వస్తుంది.
అది నెక్స్ట్ ఏమైనా మార్చుకోవచ్చు. Good morning, everybody. Really, I feel it a great privilege to share my views with our esteemed colleagues, friends, research scholars, and faculty of different departments to give lecture on the concept of plant tissue culture technique can be used for the conservation of all the plants, not only particular endangered species or threatened or nearly threatened or so, all the plants which are going to be extinct. Here, first of all, I would like to congratulate Mr. P. THIRUPATHI, lecturer, Botany, Department of Botany, Government Degree College, and his webinar chairman, M. Reddy Garu, Principal, Government Degree College, JAGTIAL, and other Vice Chairman, Masroor Sultana, Assistant Professor of Urdu, IPSC Coordinator, Dr. K. Kiran, Zoology, Off Club Secretary, M. Satyaprakash, Assistant Professor of Political Science, NSS Coordinator, Dr. M. Madhukar Rao, Assistant Professor of Hindi, Academic Coordinator, A. Shankaraya, Assistant Professor of Chemistry, Women Empowerment Cell, T. Swaruparani, Assistant Professor of Maths, and Advisor, Dr. M. Rahim. I congratulate my senior friends, Dr. T. Christopher sir, and Plant cell theory, it was slated and shown according to this theory, cell is the functional unit of life. Here to supporting this one, another one, according to Rudolf Virchow, all the new cells which are arising, they are from pre-existing cells only. If the cell is, living cell is there, then only possibility to produce new cells. If nothing is there, any non-living is there, it is not possible to produce any living cell. So here cell theory also includes a partial of Rudolf Virchow's theory, that is all living cells are arising from the concept of plant tissue culture. Haberland, he's treated as a physiologist and anatomist. And here
Slide view. Sorry,
you can change it.
share and share. Blue.
Haberland is from Germany. He is the plant anatomist and plant anatomist and physiologist. Here, plant tissue culture is based on 5 important steps. In this one, preparation of nutrient medium, inoculation, incubation, acclimatization, field transfer, and it is brief. I'll skip all these things. We'll concentrate more and more on the conservation of very different types of plants by using plant tissue culture technology. In plant tissue culture technology, laboratories should have the following equipments, particularly laminar airflow chamber, autoclave, oven, distribution unit, microwave oven, and cyclomixer. All these things are there, different pH meter, you all know because the majority of you are from degree college. And it should have the general laboratory area, medium preparation area. All these areas should be separated within the lab. This should be properly organized. You can also see the plant tissue culture laboratory should be like this. Here, within a small area also, you can create wonderful things by using this technology. Plant tissue culture technology, vessels and racks. So all the vessels and racks in large scale, you can maintain like this. Here, these are all the surgical instruments which are often used in plant tissue culture system. That is forceps, blades, holders, scalpel, etc. Surgical blades. This is the total inoculation chamber, which is having laminar airflow cabinets. So people, different scientists, they are working on laminar airflow chambers in the inoculation chamber. Here, how actually a particular plant, that is any part of the plant body is treated as the explant, which is going to be cultured. Any part of the plant which is going to be cultured can be treated as the explant. It may be leaf or anything. That is inoculation chamber. Then after that, inoculating the material, this should be incubated in the properly aerated rooms. The rooms are highly sophisticated lab with air conditioning and relative humidity and light condition. Everything has been maintained properly. Here there are plants, how they are being developed from different seeds. Here, plant tissue culture actually it includes on the fact of many plant cells have the ability to regenerate whole plants. That is nothing but 2T potency. A single plant cells without cell wall are also known as protoplasts. These protoplasts are also used in plant tissue culture technology for the regeneration of plants, and pieces of leaves, roots can often be used as to generate new plant for culture media on given requirement. Here, different types of hormones, particularly auxins and cytokines. In cytokines, we often use BAP, IPA, that is isopentanine adenine, adenine, zeatin, and other very important hormones which are going to be dissolved in different types of solvents. You can prepare stocks, etc. You can learn all these things very easy. And preparation of 2,4-D, 2,4,5-D, IAA, IBA, NA, IBA, chloram-D comba. These are all the auxins which are commonly used in plant tissue culture technology. Here, the callus cultures. Now, here, what are the different applications of plant tissue culture technique? Now, you can take any explant, you can culture on the medium. Based on the manipulation of the auxins and cytokinins, you can produce callus. For the generation of callus, generally 2,4-D hormone is used. This, when the tissue is going to be started to divide and redivide, gives an unorganized mass of tissue. This unorganized mass of tissue can be treated as the callus. From this callus, you can regenerate the plant. This callus is also useful for different purposes. Majority of the people, majority of the botanists, they think that simply the plant tissue culture technology is adopted to produce new plant, somatic embryogenesis, synthetic seeds, all these things. Not only that one, callus or calli are very useful because the callus can produce large amount of secondary metabolites which are commonly produced by a common plant. You can take a number of plants like Digitalis lanata, foxglove plant. In this plant, it has been used for heart problems. It's a wonderful medicine. It is having one particular alkaloid that is called digitalis. This alkaloid is commonly used to treat heart patients. This has been isolated from flowers as well as leaves of this plant. The availability of this particular content is very low, less from the plant, this intact plant. If you take the tissue of this one, cultured on the medium, and produce large amount of callus, bulk amount of callus, you can extract this compound in a maximum quantity. Because of that reason, today we are able to supply this type of digitalin to the pharma industry with the help of plant tissue culture technique only. Like this, number of medicines, you can take Panox ginseng. This Panox ginseng, all the slides are with me because of time constraint. I don't like to show all these things. At the end, I'll show something. Here, Panox ginseng, actually this is the plant of China. Those people, they grow this plant for about 15 years. After 15 years, they will celebrate a type of home festival in the house. They cut the plant, on that day they will celebrate. They cut the plant and take the tissues. From those tissues, they extract alkali and they add in some decoction like substance. They will take each family member will be given a cup of that decoction. They think that it increases vitality and vigor among the people. Definitely, here it is having the ginsenosides, gives very wonderful vitality in the body, will get the immunity power. Today we are suffering a lot. Recovering of this COVID-19 is more because we are Indians. We are having the great immunity power. This immunity power can be increased by using such type of medicines. One, ginsenosides of Panox ginseng. Another, Indian ginsenoside plant is Withenia somnifera. This Withenia somnifera, from the roots, we are extracting the vithalin and agenolicin. All these compounds, they are giving maximum immunity to the human beings. For that purpose, majority of the Ayurvedas, particularly what we call Chyawanprash, etc., it has been added. The compound which is isolated from Withenia somnifera has been added to the Chyawanprash, and regularly we are taking Chyawanprash, getting the immunity power. Like this callus, the plant callus can produce large amount of this. And one more life-saving drug for the cancer patients, that is vincristine and vinblastine. These two alkaloids are extracted from the Catranthus roseus plant. It belongs to the family Apocynaceae. It's called Billaganir in I think Hindi. It is a Sadabar plant. It has been everywhere. But extraction of this particular compound, the extraction procedure, it requires a lot of effort, and it will give. If you take 1 lorry of material, you will get only 1 penicillin bottle of this particular compound. That's why the cost of the medicine is very high. By adopting plant tissue culture, you take any part of its plant culture and produce large amount of callus. From that callus, we can extract many penicillin bottles, not only penicillin bottles, even cans also you can use to extract the alkaloids. That's why we are able to use this particular compound to save money. Like this, large number of many secondary metabolites which are being isolated from various plants. The same callus is also treated plants also. Suppose the plants are entered with the help of Dr. Sureka Madam. She has clearly explained about biodiversity. So India is wonderful, one of the mega biodiversity zones world. It is having large number of plants which are restricted to particular area, what we call endemic plants. And moreover, they are the medicinal plant. I have observed one very important plant, that is Decalepsis hamiltoni. These are rock areas, forest, what was acting in Annari. It's a cool drink. They add this mixture in the cool drink. That is during summer they take. They prefer more to take this one than the other cool drink like Coca-Cola also. These are all the artificial. That is the natural one. It's a wonderful taste and gives relax to the body. It is having many medicinal properties. So by using, because the plant is going to be, and moreover it is and one more thing is it is going to extinct. Either of the way, that is callus or micropropagation, and the same plant tissue, that is callus, is also used for the extraction of this particular compound. Very useful compound. Like this, the same callus is also used for the production of new plants. New plants can be regenerated by manipulating the oxygen and cytokinin ratio. Here are different types of callus you can use in callus. We have regenerated more than 6 species of plants from Cucurbitaceae family. Not only the oxygens and cytokinins, we also use casein, hydrolysate, and some amino acids like acid. By using these compounds, we can regenerate. Those papers were published in repeated journals. And these are all the organogenic callus. This callus, actually, it is going to be regenerated into shoot system. You can see here the calli may be vary. This is a nodular callus. If the callus is nodular, it is highly resistant. It can withstand the high temperature also. The skin generated easily from this. There are some examples. It is going to differentiate into plantlets. Yeah, this callus can be transported easily from one area to the other in the form of plastic bags, and the same callus can be used as a medicine and can also be in future use. NBPG are also helpful for the preservation of the calli of various endangered or other some endemic plants, which can for a longer period. Here, the direct plantlet can also be obtained because if you inoculate any explant on the medium by manipulating the hormone by using some types of acids, amino acids, and other compounds, you can also regenerate plant directly from the explant itself without intervening the callus also. Here, organogenesis, you remember always, if the only the shoot system, shoot buds are developed from the callus, this can be treated as a collagenesis. From the same callus, if the roots are directly regenerated by manipulating the hormones, particularly when the content of auxin is more, auxins are more, then the root initiation will be well. Especially till today, whatever the auxins are tested, IBA is the best one for the regeneration of roots, what we call rhizogenesis. Whereas for zeotin and the and successful for the collagenesis. Now, so the shoot tip, you can see this is the plant. Whatever the parts are there, all the parts can be used as the explant. Yeah, shoot, but how actually from the callus, centrally located callus is there, from the callus, gradually there is the differentiation of shoot buds. Here, shoot bud differentiation in Cassia angustifolia. One more important plant, Cassia angustifolia, that is nothing but Surnamukhi leaf, which has been used to control the constipation. What we see here, Kayam Churna and other number of drugs are available, powders are available. All the powders are prepared by using the leaves of Cassia angustifolia. Wonderful leaf. It is better to have at least once a month for free motion and relief from the constipation. The same plant has been regenerated by using different types of explants. Here, lateral bloods from the callus can also be issued. You can see that the main advantage of plant tissue culture is if you take any okay, here multiple shoots can be produced by using this one. Here, one plant is very rare plant which is growing in not in India and Australia area. That is St. Polio Ionantha. This is a flowering plant. This has been regenerated by using plant tissue culture technology. These plants are very rare. And single shoot elongation, how actually a single shoot is also elongated and developed directly by using cytokinins, etc. Gibberellins. and complete plantlet can be seen in a beaker. Here in vitro plantlet formations you can see in the test tubes. Here at any plantlets, the very small plantlets you can see. Yeah, pear tree plantlets, you know, Pyrus communis, pear plant, they regenerated. Now I'll go for the another very important application of plant tissue culture, that is somatic embryogenesis. Here, generally embryos are nothing but they are derived from the zygote. How zygote is formed? By the fusion of male and female gamete, zygote will be formed. The zygote undergoes repeated division, gives rise to embryo. But whereas in tissue culture, whatever the somatic tissue is inoculated, this somatic tissue directly, sometimes it may give callus or via callus, sometimes it may be differentiated embryo-like structures, heart-shaped structures. You can see here, these are all the structures. These somatic embryos, the greatest ability of the somatic embryo is that each somatic embryo is able to directly develop into new plantlet. Thus, the plantlets can be directly developed from this one. Here it is nothing but a canola, it is nothing but a Brassica napus. In some plantlets which are directly developed from callus stage, somatic embryo to plantlet to plantlets. Here I want to explain very, very important application of plant tissue culture. Number of rare plants are there, very rare plants are there, endemic plants are there, endangered plants are there. In such cases, one beautiful example is Macapuno variety. It is commonly growing in Maldives and Myanmar areas. This coconut variety is a wonderful variety. It has a very good amount of coconut, sweet to taste, and the coconut can be stored for longer period. But the plant, by you take seed and sow in the soil, you won't get plant. The reason is, in almost all the seeds, what we call fruits, you won't find embryo. The reason is, at early stage, that is immature condition, the embryo will be departed, that is aborted. When the embryo is aborted, is there a possibility of the development of new plant from this one? No. That's why in such cases, you identify that plant. When embryo is set, with the help of instruments in the laboratory, you pluck, you cut the fruit and take into the lab and open it. You can identify the embryo. That immature embryo can be cultured on the medium by manipulating the hormones, by using different types of media like MS, LS, all these media are there. So here you can produce new plants. This is the only possibility of this particular production of a rare plant. That is the Macapuno variety of coconut. This type of large number of fruits are there, size is more, taste is good, and moreover it can be preserved for longer, but the plant is very rare. This can be done. Here embryo, these are the embryos which are very smaller in condition. They generally they are present in between the cotyledons or in the endo, sometimes present in the embryo, etc. You can see the embryo of potato, you can rescue it and sometimes transformation of microspore derived and zygotic embryos of wheat. Here, sometimes you can also produce some embryo-like structures from the what we call anther culture also. Here, ovule culture. This is one of the, here I want to tell one more application in embryo culture. Many plants are there. You can take one variety, slide is not here. In normal method, you know well, Solanum tuberosum and Lycopericinum esculentum. One is tomato, one is potato. Both belongs to same family. Our scientists, they tried to produce a single plant which should give the underground potatoes and aerial tomatoes. That was their objective, breeder's objective. For that purpose, they tried in between tomato and potato, but it is not possible because of various incompatible barriers. Whereas the plant tissue culture method, it gives a very good option by using protoplast fusion, what we call somatic hybridization. By using somatic hybridization, but just you take these two cells from the leaves and remove the cell wall with the help of enzymes like cellulases, pectinases, acidozyme enzyme. You can remove the cell wall, and these two protoplasts can be fused and gives rise to a heterokaryon, what we call from that heterokaryon, you can raise the callus. From the callus, plants can be produced. Those plants, they can produce the aerial tomatoes, undergo potatoes, that is called tomato plant. A number of names were given to this also. That is protoplast, application of protoplast, somatic embryogenesis. One more is ovule culture. Here, ovule is nothing but it is a female part. After fertilization, ovule develops into seed. Before fertilization, in some cases like cotton, you might have seen in cotton, the cotton fibers are developed only on the seed. If you want to, some varieties, good variety of cotton is there, which is giving good quality of cotton. You take the ovules of that one and culture directly without intervening the fertilization. If you culture the ovule, those ovules directly produce this cotton. It's a purely natural one. Cotton is very soft in nature. It has been used in textile industry. This is one advantage of ovule culture. It is application of one of the important applications of plant tissue culture. Here, yeah, triploid papaya by endosperm culture. You can see endosperm is the product of fertilization by the fusion of three important cells, one male gamete, two or nuclei, what we call secondary cell, all three together fuse and gives rise to a parent. Helpful is also used as an explant for the production of triploid plant. Why we are very much interested to produce triploid plant? In some cases, like papaya and other plants, the triploid plants are having number of advantages. The size of the fruit is more, and the quality of the pulp is more. The number of seeds are also less. Because of that reason, you can culture the endosperm. You can also produce a triploid papaya plant by endosperm culture. It is not at all possible in a normal condition to produce triploid plant. Only the plant tissue culture is giving an option, helpful. And in addition to this one, another one, soma-clonal variation. You know well, the term itself indicates soma means somatic cells. Clonal means all the cells, they are derived from the single parent. Clone is nothing but group of cells which are derived from single genetic trait. When an explant is inoculated on the medium, that explant undergoes repeated mitotic division. During the course of mitotic division, rapid mitotic division, because of various reasons, sometimes cell wall may not be formed because of endoduplication. The chromosomes may remain within the cell. The number of chromosomes will be doubled. Because of that reason, those tissues, they show variation. That is called soma-clonal variation. If you raise the plants from these soma-clonal variants, they are so useful in plant breeding program. Number of plants, for example, even today, some citrus species, some mango species are developed by using soma-clonal variation. The variation was, the term was first, you know, well, anthers lobes are the main male portions. When you cut the anther, you will get microspores. Instead of cutting and doing this one before anthesis, what we call anthesis, opening of flower. Before opening the flower, you take the anthers and culture on the medium. They will give rise to plants. What plants? Haploid plants. Here, one can produce directly haploid plants by culturing anthers, because the microspores, they undergo repeated division, gives rise to callus. From the callus, regeneration will take place. These haploid plants are having many advantages in breeding program. Suppose you take when a particular cross-pollinated crop is there, in this one, the fruit size is measured, everything is measured. Actually, the size of the fruit is it coming the character from mother side, means female side or male side, or etc. can be studied. For that purpose, these haploid plants are very, very useful. Direct haploid plants are totally sterile. They can be diplotized by using, you know well, that is colchicine. When you treat this one, colchicine, the haploid tissue will be diplotized. From those diplotized, the plants will develop. From those plants, you will get pure type of varieties where you can find the quality of, you can test, you can study the quality of fruit, quality of kernel, etc. can be studied. Yeah, protoplast, I have given one example already. And protoplast, brassica protoplast, very small. Always there is a great demarcation between plant cell and animal cell. In plants, the plant cell is surrounded by plant cell wall, whereas in animal cell, you cannot cell wall, because the plants which are growing outside, they are withstand a different type of adverse condition because of the presence of cell wall only. Otherwise, they cannot tolerate. See here, the last 4 or 5 days back, the temperature was very high, up to 47 degrees centigrade. But plants can withstand, but we cannot withstand because they are stationary condition. Because of the presence of cell wall, they can tolerate all the adverse conditions. Here, the one more promising area of plant tissue culture is cryopreservation, gene banks. Already, Dr. Christopher sir and Madam ex situ in situ conservation, in those cases, they are clearly explained how one can store the plant material in at low temperature by using liquid nitrogen, that is at minus 196 degrees centigrade. So if you visit Sweden, those people, they developed gene banks, underground gene banks are there to avoid all the natural calamities. However, natural calamities occur with gene banks since they are underground. That type of foresight they are having regarding the preservation of germplasm preservation. You know all these things. Here, gene banks, you can see some gene banks which have been maintained in different corners of the world. But in India, we are actually very unfortunate. We are not having this type of, but NBBGR, it is doing excellent job from last decade. Yeah, tissue culture racks with screw bottles and all. Yeah, after these plants, whatever the plants we have raised, those plants can be rhizogenesis can be done by using IBR. So those plants can be transferred into the field by intervening the what we call acclimatization system. After acclimatization, they can directly develop into transferred into the fields. Yeah, these are all the different plants. Here you can see large number of plants. I think majority of the people, they know about stevia. The stevia is a sweetener, natural sweetener, not energy giving one. Market lo you know well. This also cultured and plants can be produced. You can also grow from Sima plants are available. Get it, and you can grow in our laboratory also. Orchid tissue culture is another promising area of plant tissue culture. Getting income from this source, particularly Bangalore, India Institute of Horticulture Institute. It is producing large number of orchids by using plant tissue culture only. You might have seen the decoration of marriage platforms, areas with the help of orchids only. All those orchids are produced by plant tissue culture only. The normal method, you cannot produce such type of within short span of time. Here, micropropagation is nothing but large amount of plant production. These are all the slides you can see. It's a very promising micro banana. Banana micropropagation is also one of the wonderful area where you take one banana quality. If you want to produce large number of bananas, bunch of bananas, you select the plant and from the same plant, explant is taken and culture produced. Whatever the bananas today, they are available in the market. They are sweet to taste, larger in size, because all they are tissue culture raised bananas only. Some other plants. Geranium. These are all the flowers in floriculture, flower industry, all the sectors. Plant tissue culture is playing prominent role, but people are not utilizing this area, this technique properly. Labs should be established properly. Always we try to develop the new plants. We should identify the endangered endemic plants and collect those plants and cultured in the laboratory and develop those plants. In vitro fertilization is also another promising area. Here, if you are able to produce the flowers in a test tube, that is called in vitro flowering. If the plants are producing flowers within the test tube, you can also develop an in vitro fertilization system. For various reasons, you can develop highly desirable varieties of plants. In vitro, yeah, there is a rose in vitro flowering. Directly, you can produce the rose flowers in the laboratory itself. This is the one of very advantageous aspect of plant tissue culture. Banks, all those are there, but next, yeah, before I want to conclude this one. Next, before I want to conclude about this biotechnology, that is plant tissue culture technique, which is an ideal for the conservation of rare and endemic endangered species which are going in India and around the India, that is in world. Plant tissue culture should be used nowadays. Banje. Yeah. Before concluding my on వస్తుంది కదా, క్లియర్ గానే ఉంది కదా. Before concluding my lecture on the as the application of plant tissue culture for the conservation of plants, I want to tell you a message to all the participants here. Angiosperms are vary, they are different with respect to size, shape, habit, nutrition, inflorescence, flowers, etc. The same angiosperms are providing food, shelter, medicine, fibers, gums, tannins, latex, alkalides, etc. for the welfare of human being before and no and very small, just 0.5 centimeters. They too belong. Angiosperms, you can see, belongs to angiosperm, and largest angiosperm, this one left side, not the right one, that is a Sequoia sempermensis, belongs to gymnosperms, where you can see in California city. Eucalyptus regnans, it grows up to 120 meters height. Here variation, largest leaves bearing the angiosperms are nothing but Victoria regia and Raphia tedicera, which are size of leaves are larger. Here even in some areas, usually when they go for bath, they keep their children on the leaves, so to protect from various animals or so. And largest inflorescence, you know, Amorphophilus titanum, which is about the up to 6.5 feet height. Here the smallest flower you can find in lemna, and largest flower Rafflesia. Why I'm telling all these plants, they belong to angiosperms only. The smallest seeds are found in Wolfia angustia, and these are all the fruits. The largest fruit, it is pumpkin, belongs to our family, that is nothing but Cucurbitaceae. Yeah, I'm working on Cucurbitaceae. Recently we could identify by using the markers at the sex of the plant at early age in Momadica dioica. And smallest and largest seeds, you can see epiphytic orchids. Orchids, they belong to microspermae according to Bentham and Hooker's system of classification series. Microspermae are nothing but very smallest seeds. The seeds are just like particles, but they are having lot of amount from this one. And Lodacia maldivica, it belongs to Palmae, what we call Ericaceae family, the largest family, the largest fruit. Here orchids, you might have seen all the orchids, wrapping plants, etc. Here large number of scientists, because there is a great variation with respect to plant size, shape, flowering, etc. Number of medicinal plants like this, all these are some endangered, endemic medicinal plants are there. All these plants have been started to conserve by using the plant tissue culture technique. Here the last my message is always, as the botanist, as the plant scientist, you give maximum importance to taxonomy. It is vanishing. People are neglecting the taxonomy. With the help of that, you can identify the plants. After identifying, we can do whatever we want. We can create. So for that, I want to tell, that is identify and conserve and know about the medicinal importance of that particular plant in your area. Thank you. Thank you for giving me this opportunity, particularly the organizing committee of Government Degree College JAGTIAL, especially to the principal M. Srinivas Reddy Garu and his team, and our coordinator P. Tirupati for giving me this opportunity. And if you want any slide related to this one, plant tissue culture, other slides, number of slides are there. I'll send to Tirupati. He will share to you. You can get it. Thank you. Thank you.
Okay, sir. Thank you very much, sir. You are giving such a wonderful session and excellent information. We are gathered here. We learned so many things, new things about the conservation of biotechnological view and in the tissue culture and conservation and new ideas we learned. For this session, we are very, very thankful to you. We are very enlightened your session. Thank you very much, sir. On behalf of Government Degree College for Women, JAGTIAL, we are very thankful to you and give such a wonderful lecture. And you are the last lecture from this for this national webinar. And you are very busy schedule. You give a wonderful time for us, and we most enlightened. Our friends, most of our friends, our participants are belongs to degree college faculty, and some of the participants, they are belongs to different universities of the esteemed universities from different states of our country. They are we hope they are very enlightened, and they reach the topic we choose, the biotechnological applications for the conservation of biodiversity. They are all the participants, they are very enlightened from these three lectures. Dr. M. Sureka Madam Garu and Dr. Muhammad Mustafa sir and Dr. T. Christopher Ruben sir. They are the three esteemed eminent professors of Kakatiya University, Warangal. We are very, very thankful to you, and thanks a lot, sir. Before we conclude, we invite our esteemed principal, Malaram Srinivas Reddy sir garu. Please give a message about this webinar. Sir, mute lo unnaru. Srinivas Reddy sir, you are in mute.
Okay, thank you. Thank you, Tirupati garu. Once again, I am very thankful to all the dignitaries and participants who made this webinar today very grand success. And I once again I convey my sincere thanks to our eminent resource persons, Dr. Sureka garu, Dr. Mustafa garu, Dr. Ruben garu, who given more valuable lesson and more valuable feedback and more valuable knowledge to all the participants. And I once again congratulate the Department of Botany and a team of JAGTIAL who made very good efforts to conduct and made this webinar grand success. Once again, thank you all. And over to Tirupati sir, please.
Okay, thank you very much for giving your continuous support and encouragement for conducting this national level webinar. We hope, we expect from you these type of encouragement and motivation continuous for further also. We are very thankful to you on behalf of Department of Botany and our participants. From our participants, we are very thankful to our esteemed principal, sir. Now, I would like to thank you and each and every participants of our country to give such a valuable time and learn more things or more new aspects and more new knowledge from our esteemed lecturers of our Kakatiya University, Warangal. And we are very, very thankful to them because of this is a very pandemic situation. They are giving valuable time for us and they are more enlightened for us and they are giving more valuable time, more than 3 hours. We are very, very happy and thankful to them. And also, we are very, very thankful to you, especially Resonance IIT NEET Academy Warangal, because of the Zoom our Zoom is only accepting only 100 participants. But this Zoom, Resonance Academy IIT Warangal, they are giving platform, such a wonderful platform. More than 1,000 of the participants, they are participated in this webinar. That's why we are very, very thankful to them, because of this is a very critical period for giving their IIT and NEET coaching time, but they are alternately arranged their students webinar and giving this platform for us for enlighten our different degree colleges lecturers and also different university professors. Because of this is a wonderful session, and we are very, very happy, and we are very happy to share with you, because of one person is continuation support for this webinar. That is our beloved brother, Assistant Professor of Botany, SRR College, Karimnagar, Prasanna Harikrishna Puli, Tiger Prasanna Harikrishna, my beloved brother. Because of yesterday afternoon, very, very critically accepting and giving this platform for us, because of every corner of the nation, different professors, different university lecturers, they are participated in this webinar. That's why he is insisting them and requesting them and convince them and cooperate with us. And our Prasanna Harikrishna is there, simply is there. The tiger is sitting there in front of us. Prasanna Harikrishna. Okay, we are seeing them. Okay, seeing her, seeing his photo, and is online also. And on behalf of Government Degree College for Women, JAGTIAL, we are wholeheartedly thankful to you, my dear brother, Prasanna Harikrishna sir, convener, Career Guidance Cell, SRR College, Karimnagar. Thank you very much. Thanks a lot. And we have my sincere thanks to my department of Botany, Government Degree College for Women, JAGTIAL, my colleague, Srimati Sandhyarani Banda, and our senior faculty, Vice Principal of our college, Dr. Masroor Sultana Madam Garu. Dr. Masroor Sultana Madam, she is more senior faculty member of our college, and she is most encouraging me for this conducting of this webinar. Dr. Masroor Sultana Madam Garu, Vice Principal of our college, Assistant Professor of Urdu, and M.A. Rahim sir. M.A. Rahim sir, Assistant Professor of History in Urdu, is a very senior faculty of our college, and he called me as a brother. But I am a very, very junior, very little in front of him. But his continuous encouragement, we are very, very thankful to him. And our college academic coordinator, Sri Amballa Shankaraya sir Garu, for his continuation encouragement, and staff club secretary and task coordinator of our college, my brother, my beloved brother, Mamidipelli Satyaprakash sir Garu. He is elder son of my guru, Rajana sir, and IQAC and NAC coordinator, Dr. K. Kiranmay Madam Garu, Assistant Professor of Zoology, for his motivation and encouragement. We are very, very thankful to her. And the NSS coordinators of our college, Dr. Mahindrakar Madhukar Rao Garu, Assistant Professor of Hindi, and second unit, NSS unit, Srimati A. Rajini Madam Garu, Assistant Professor of Physics, for their continuation, motivation and encouragement for this conducting of this webinar. And also my brother, Dr. Tota Mahesh, Assistant Professor of Zoology, is working as a Assistant Professor of Zoology in SRR College, Karimnagar, but now he is working as a OD base in GDC Women, JAGTIAL. And GVR Sai Madhukar sir, he is also Assistant Professor of Chemistry in our college. And my sincere thanks to our library in charge, మా టీవీ ఇన్ఛార్జ్ అసిస్టెంట్ ప్రొఫెసర్ ఆఫ్ తెలుగు రాపర్తి శ్రీనివాస్ గారు, and my beloved brother JKC Tirupati, and my beloved brother computer faculty Satyam sir, and English department D Sunita, head of the English, Srimati D Sunita madam garu, and E Josna madam garu. Economics department V Jamuna madam garu. History department Padavi madam garu, and commerce Mamata madam garu, and computer Sangeeta madam garu, and Urdu Arshia Sultana madam garu, and Yasmin Sultana madam garu, and non-teaching staff of our college, each and every staff member, non-teaching staff member. On behalf of our botany department, we are very thankful to them for their continuation support and motivation for conducting this webinar. And each and every supporting member, we are very, very happy and thankful to them on behalf of this conducting of this national webinar on biotechnological applications for the conservation of biodiversity. We hope this seminar is more enlightened because of very esteemed university professors, eminent professors. Our 3 professors, they are giving such a wonderful session. We are very happy, and each and every participants, we are wholeheartedly thankful to them. Thanks a lot. We have already sent feedback from our message chart. Please fill the chart, fill the feedback link. Click on feedback link and fill the feedback form, and you will get e-certificate within a couple of days. Please fill it before leaving this session. And each and every participant, we are very, very thankful to them. Thank you very much.
Hi, Tirupati Garu. Congratulations for organizing such a beautiful webinar.
Valinath sir, we are very, very thankful to you, sir, because of your, we are meet at Maulana Azad University.
Thank you, sir.
And orientation training. You are the most senior faculty of chemistry from Gunturu. Thank you, sir, Valinath sir.
Thank you very much. Thank you for the opportunity.
వెంకన్న నిజామాబాద్ పోయినట్టు ఇవాళ ఎస్డిఎల్సి పని మీద నిజామాబాద్ ఎస్డిఎల్సి పని మీద అట్లా కదయ్యా ఆల్టర్నేట్ అరేంజ్మెంట్ చేసుకుంటారా లేకపోతే ఎవరు ఇది ఉంటారా ఆయన చెప్పి వద్దే వద్దు ఏ తిరుపతి వద్దు
యాక్చువల్ గా ముక్క దగ్గర ఉంచి ఇందరిని ఫెలిసిటేషన్ చేసి వాళ్ళకి చూపిద్దామని అనుకున్నాను.
Leave the meeting and across.
Thank you, Tirupati sir.
Thank you very much, sir.